Good evening, everyone. Good evening, everyone. Whether I'm audible? Yes, good evening, good evening. Good evening, everyone. Yes, after so long. I hope everyone has completed your syllabus. Okay, yes, I hope everyone has completed the syllabus. Okay, we have 6 revision class. Okay, total 6 class will be there. Yeah, total 6 class will be there. My kind suggestion, you have to attend all the 6 class. Okay, I will assure you, if you attend our marathon series, this is quick revision series. If you attend, minimum 50 to 60% of questions will be covered from this syllabus only. Okay, so you be attentive in the class. If you have any doubt, kindly ask me there itself. Okay, today we are going to start with plant biotechnology syllabus. Okay, particularly we are going to see about plant biotechnology. This is our syllabus. Okay, in this class today I am going to cover about genetic engineering, restriction enzymes, vectors, gene cloning, then gene transfer methods, apart from the transformation, okay, recombination, heterosis, general application of biotechnology. Okay. Apart from that, I am going to cover some of the concepts of bioinformatics, genomics and proteomics. Okay. So entire biotech syllabus, okay, we are going to cover in only 2 sessions. Okay. In 2 sessions, we are going to cover entire biotech syllabus. Okay. So you know the importance. Okay. Revision is one of the most important thing. You just listen the class properly. Okay. Whatever doubt is there, you just ask me there itself. Okay. First, we are going to start with genetic engineering. Okay, genetic engineering. This we already know. Okay, genetic engineering means what? It's a deliberate modification of an organism genetic material. Okay, we will modify the genetic material of the organism. Okay, by addition of the gene or deletion of the gene. Okay, simply we can say modification of the DNA sequence. Okay, this genetic engineering we can also call as what? Recombinant DNA technology. Okay, this is another important objective point. Okay, recombinant DNA technology. Why, sir, we have to learn about recombinant DNA technology? Okay, the central goal of this recombinant DNA technology is some of the heritable traits that are not arise through conventional breeding can be covered in this recombinant DNA technology. Okay, this is the purpose of studying about genetic engineering. Okay, even some of the heritable traits that are not arised through conventional breeding can be obtained through this genetic engineering. Okay, this is the basic definition. This definition they have asked in 2019 JRF question. Okay, those who have attended the previous year question paper sessions, they know about this. Okay, so we are going to see about the scientist contribution also. Okay, the most important thing, Paul Berg, he is the first person created the recombinant DNA molecule. Okay, rDNA molecule was developed by Paul Berg. Here also very important, 1972. Okay, first genetically modified organism. Okay, the difference is clear now. First recombinant DNA molecule was developed by Paul Berg, and the first genetically modified organism was developed by Herbert Boyer and Stanley Cohen. This is Very important objective question, and it is one of the repeated question. Repeatedly they have asked about this. Okay. First GMO organism was developed by Boyer and Conson. Okay. Then the most important thing, first genetically modified crop approved for commercialization. Okay. That is flavor saver tomato. For what purpose? What is the purpose of developing flavor saver tomato? This we have seen in our class. Comment your answers. Flavor saver tomato, what purpose? Option A, for increasing shelf life. Option B, increasing TDS. Option C, enhancing the color of fruit. What will be the answer? Why the flavor saver tomato has been developed? Yes, I am expecting answer. Yes, very good. Increasing the shelf life. Okay, flavor saver tomato. This is the first commercialized GMO crop. Okay, you have to remember one thing. First developed GMO crop is tobacco. Okay. Tobacco is the first developed GMO crop, but first developed GMO crop that is for approved for commercialization. That is flavor saver. Okay. You have to know this difference. Okay. In question, what they are asking, you have to read the question, then you have to answer. Okay. So we have developed a genetically modified organism. Okay. This approval for commercialization will be given by GEAC. Okay. This is the nodal organization. Genetic Engineering Appraisal Committee. Okay. This is another important objective point. Okay. GEAC will approve. Okay. This will approve the commercialization of the GM crops. This will comes under the Ministry of Environment. Okay. Ministry of Environment and Forest. Okay. This is another important point. Okay. Now we are going to see about DNA cloning. Okay. This everything you know. Okay. DNA cloning means what? It's a process of producing multiple identical copies of a specific DNA sequence. This we know. This is the DNA cloning. Cloning means was producing the multiple identical copies of a specific DNA sequence. How? By inserting it to a vector and propagating within an host organism. This we have already seen. Okay. These are the basic steps. First, the desired fragment should be isolated. Then that has to be cloned in cloning vector. This vector containing the gene of interest will be introduced into the host cells. Okay. In the host cells, this will be replicated. Because of that, multiple copies of the selected fragment will be produced. Okay. This is the basic step we have already seen. Okay. See this diagram. Vector will be there, that will act as a cloning vehicle. Our DNA fragment will be there. This will be inserted in the vector. This will be transformed to the host bacterium. In bacterium, this plasmids will multiply. Okay. Because of that, identical copies of our target gene will be produced. Okay. This is just a basic outline. Okay. One time in 2021, they have asked the sequence. Okay. First, what we have to do? Okay. Like this sequence they have asked. Okay. We are going to see all the things. How to select the recombinant from non-recombinant cell. Everything we are going to see in our class today. Okay. If we are going to do DNA cloning. Okay. Some of the basic requirements are needed. What are the requirements? First, restriction enzyme is needed. For what purpose? For cutting the gene of interest as well as cutting the vectors. Okay. The purpose of restriction enzyme is for isolating or selecting the gene of interest from the genomic DNA as well as cutting our vector to insert our gene of interest in vector. Okay. Apart from that, several modification enzymes are needed. Okay. DNA ligases is needed for joining this vector with gene of interest. Cloning vectors, this also needed. Then host organisms like E. coli is needed. For what purpose E. coli is needed? Our vector will be inserted in the E. coli. This E. coli will be multiplied. So our target gene will also multiplied. Clear? Up to this clear? Any doubt is there? Any doubt is there? I need interaction. Just you message clear or not? Yes, if clear means I will proceed next thing. Okay. Yes. See, these are the different range of DNA manipulative enzymes are there. Okay, for example, nucleases, ligases, polymerases, yeah, polymerases and modifying enzyme. Okay, what these nucleases will do? These nucleases will cut, shorten or degrade the nucleic acid molecule. Yes, nucleases will cut the fragment of DNA, or it will shorten the segment, or it will degrade the nucleic acid. Okay, ligases, what it will do? It will join the different fragments. Polymerases, it will make copies of the molecule, and there are different modification enzymes are there that we are going to see in our upcoming sessions. Okay. The most important thing for exam point of view is you have to know if they have given some of the enzyme, what will be its function, what are the different DNA modification enzyme, and what it will be, and what it is functioning in the cloning experiment. Okay. For example, exonuclease are there. There are two different classes of nucleases are there, exonuclease and endonuclease. Okay. Exonuclease means what? It will remove the nucleotides from outside of the DNA molecule, at the end of the DNA molecules. Endonuclease means it will cleave within the DNA molecule. This is clear. So here the most important thing we are going to discuss, what are the examples of exonuclease and examples of endonuclease. This is most important thing. This is most repeated question. BAL 31 is an typical example for exonuclease. Okay, this is most repeated thing, BAL 31. Okay, the most and the specialty of this BAL 31 is it will remove the nucleotide simultaneously from 3' end and 5' end. Clear? This is another specialty of this BAL 31. Clear? Another example of exonuclease is exonuclease 3. These are the example of exonuclease. And coming to the endonuclease, S1 endonuclease, DNase, these are endonuclease. And the most important thing, restriction endonuclease is a class of endonuclease enzyme. Okay, this is very important enzyme, restriction endonuclease. Several times they have asked much question from this topic. Okay, how we are naming the restriction enzymes. Okay, what is the characteristics of restriction enzyme? Whether it is a 4-base cutter, 6-base cutter, like that, different questions they have asked. Okay, this also we are going to discuss in our section. Okay, then another class, DNA ligase. As we already discussed, DNA ligase means what it will do? It will ligate the DNA fragments. Okay, it will ligate the DNA fragments. Okay, joining the two molecules will be done, or else the discontinuity, that means missing phosphodiester bond between the nucleotides, will be joined by DNA ligase. And most important exam point of view is DNA ligase cofactor. This you have to remember. DNA ligase cofactor. If E. coli means the cofactor for DNA ligase will be NAD molecule. This is very important objective point. Okay. T4 DNA ligase. T4 DNA ligase means it will use cofactor ATP. Clear? This is most important and repeated objective question. Okay. What are the cofactors required for DNA ligase enzyme? If it's an E. coli enzyme, that will require NAD. If it is an T4 DNA ligase enzyme, it requires ATP as an cofactor. Clear? Next, coming to the next class, polymerase. Okay, DNA polymerase enzyme. We know what the polymerase will do. It will synthesize the new strand by using the primers with the existing template. This is the general definition of DNA polymerase enzyme. Okay. In the polymerase, if you see, two different types are there. First, you see clearly here, DNA polymerase 1 and Klebnow DNA fragment. These two are very important question, and they have asked this one time in assertion and reason type. Okay. Now we are going to see DNA polymerase, what it will do. It will polymerase the synthesis of nucleotides. Okay. See here, some missing nick is there. This nick was synthesized here, and the most important thing, for proceeding to the nick, the existing nucleotide was also replaced here. This is the question they have asked. If we use DNA polymerase 1, even the existing nucleotides will be replaced. Okay? But if we use club now fragment of DNA polymerase, the existing nucleotide will not be replaced. Clear? Why it is not replacing? That also we have to know. Why it is not replacing? If you see the DNA polymerase 1, it will have three different activities. This is, we already know, 5' to 3' polymerase activity will be there. 5' to 3' exonuclease activity will be there. And 3' to 5' exonuclease activity will be there. Okay. This club now fragment of DNA polymerase that will lack the 5' to 3' exonuclease activity. That's why the existing nucleotide are not replaced. I hope this is clear for everyone. Okay. This is most important point. Okay. Next question. This they have asked in match the following. Okay. Different modifying enzymes and what is its major function. Okay. Alkaline phosphatase, the name itself is there. Phosphatase, it will remove the phosphate group from 5' of the DNA. Okay. Remove the phosphate group from 5' of the DNA. Polynucleotide kinase. Kinase, what it will do? It will add phosphate group. Yes, it is adding phosphate group at 5' end. Then another most important thing, terminal deoxynucleotide transferase. Okay. What this enzyme will do means it will add nucleotide to the 3' end of the existing double strand DNA molecule. Clear? That the enzyme is terminal deoxynucleotide transferase. I hope this is clear. So, different enzymes we have seen that are used in molecular cloning. Okay. Another important thing, restriction enzymes, I already told. Okay. Every year they are asking minimum 2 to 3 questions from restriction enzymes. This is very important topic. We are going to see how the restriction enzymes are named, what are the different classes of restriction enzyme, which class we will use for this cloning purpose. Okay. And some of the important restriction enzymes also we will see here. Okay. So restriction enzyme, the name itself is there. Okay. Restriction enzyme. The main function of the restriction enzyme is what? It will cleave the double stranded DNA into different fragments. Okay. Double stranded DNA into different fragments. Okay. If you see the concept, if a double stranded DNA is there, there will have a recognition site. This each restriction enzyme will have a particular recognition site. Okay. After recognizing this particular site, this will make double strand break. Okay. This will make a double strand break. This was primarily discovered in bacterial species as a mechanism of innate defense. Okay. If you think, then how these enzymes, then how the bacterial DNA is protected from this enzyme? That also we have to know. If the bacteria will have, if the bacterium is having the restriction enzyme, it will also have a DNA molecule. How this DNA molecule is protected from this restriction endonuclease activity? You see the cell, the plant, sorry, these cells are very clear. Okay. What it will do means, wherever the recognition site is there in the DNA molecule, this recognition site will be methylated. Clear? Wherever the recognition site is there in the DNA molecule, that will be methylated. If it is methylated, this restriction enzyme will not recognize the site. Because of this, the cell's own DNA molecule will not be cleaved. I hope this is clear for everyone. Okay. Who has discovered this restriction enzyme? This they have asked one time. Okay. Werber Arbor, Daniel Nathans, and Hamilton Smith. These are the three scientists they have discovered this restriction enzyme, and for this work they have got Nobel Prize in 1978. Okay, Nobel Prize in 1978. Another most important thing, which is the first discovered restriction enzyme, TK, Hind2. TK, this is the first discovered restriction enzyme, TK, Hind2. TK. Now we are going to see how we are naming this restriction enzyme. Why, sir, Hind2? Why we are naming, how we are naming the restriction enzyme? If you see, the major restriction enzyme is EcoR1. Okay, EcoR1. If you see here the naming system, E, this is the first letter of the genus Escherichia. Clear? Escherichia, first letter of the genus. Then next two letter will be species name. Okay, Eco. Okay. Then followed by its strain and followed by its order of discovery. Okay. By this they have named the different restriction enzyme. For example, here BAM-H1, Bacillus amyloliquefaciens, strain is H, and this is the first restriction endonuclease isolated from this bacterial species. Okay. So that's why they have given the name one BAM-H1. Okay. In statement question, they can ask, restriction enzymes are also present in bacteriophages, whether it's true or not. Restriction enzyme is also present in plants. Restriction enzymes are present only in bacterium. What will be the answer? What will be the answer? Restriction enzyme is present everywhere, present only in bacterium. Yes, what will be the answer? Restriction enzyme will be present only in bacterium. Okay, only in bacterium. That too, it's an what? It's an innate immunity mechanism. Okay, innate immunity mechanism. Okay, clear. Next, if you see the recognition sequence of this restriction enzyme, this will be always palindromic in nature. Okay, this is palindromic in nature. These are the key points they will ask in the question. Okay, these are palindromic in nature. Okay, for example, EcoR1, this is 2019 objective question. Okay, EcoR1, the recognition site is 5' G AA TTC. Okay, so like this it will be there. Okay, it's a palindrome. If you read from 5' to 3 direction, the sequence will be similar. That's why we are saying it's a palindromic sequence. Okay, then this restriction recognition site will be highly specific. Okay, and this restriction enzymes, the recognition site, and after that it will cut. That may be either 4 to 8 base pair sequence will be done. That's why we are saying it's a tetrabase cutter, hexabase cutter, or octabase cutter. Okay, tetrabase, hexabase, or octabase cutters, like that we will say. Okay, this is very important point. They have asked one time in EcoR1, what is the recognition and restriction site? This is the site G 5' G AA TTC. Okay, this is the recognition site for the restriction enzyme EcoR1. If it is recognized and it will cleaved, it will produce sticky fragment. This is also repeated question. Okay, EcoR1 will produce sticky fragments after cleaving. Okay, some of the restriction enzyme will produce blunt ends. Okay, blunt ends after restriction. The typical example is BAM-H1 or ALU. Okay, typical example is ALU. Okay, this is the typical example that will produce blunt ends. Okay, after restriction. Okay, then the most important thing, the frequency of the recognition site. This they have asked in 2019. Same I am saying. 4 questions they have asked. This is the formula. Just you remember the formula for calculating the frequency of recognition site in a DNA molecule is 1 by 4 power n. Okay. If it's an tetrabase cutter, n equal to 4, 1 by 4 power 4. Okay. If it's an hexabase cutter, 4 power 6. If it's an octabase means 4 power 8. Like that we have to calculate. So what it is saying means if our DNA is 100 MB in size, okay, 1 in every 256 nucleotide, this may go and cut the site, like that meaning. Clear? So the formula for finding the recognition sequence in a DNA molecule is 1 by 4 power n. I hope this is clear for everyone. 1 by 4 power n. Okay. You see here the typical example I told, ALU1. It will produce blunt ends. Eco R1 will produce sticky ends. Okay. These are some of the other restriction enzyme that are commonly used in gene cloning experiments. Okay, clear? I hope this is clear for everyone. Okay. Now coming to the classification. There are three different classes of restriction enzymes are there. Okay. Type 1, type 2 and type 3. We are going to see the important things only in exam point of view. Type 2 restriction enzyme is commonly used in gene cloning experiments. Okay. Type 2 restriction enzyme. The specialty of this type 2 restriction enzyme is it will require only magnesium as a cofactor. Okay. But if you see type 3 and type 1, it will require ATP and magnesium and SAM and ATP, magnesium and SAM. Okay. So the specialty of type 2 restriction enzyme is it requires only magnesium as a cofactor. Clear? And most important thing, the recognition site and restriction site both will be happen at the same site. Okay. It will recognize and it will restrict at the same site. Okay. But if you see in type 1 restriction enzyme, it will recognize one site, then away 1000 base pair away from the recognition site, it will cleave. The concept is clear. So from the recognition site, around 1000 base pair away, it will cleave. Okay. Then if you see type 3 restriction enzyme, 24 to 26 base pair away from the recognition site, it will make the double strand breaks. Clear? And the most important and repeated thing is example of type 1 restriction enzyme and example of type 3 restriction enzyme. This they have asked several times. Okay. Eco KB is a typical example for type 1 restriction enzyme, Eco K and Eco P. Okay. And for type 3, Eco P1 and HINF3, these are the typical example for type 1 and type 3. This is most important thing. Okay. Now we are going to see the different classes of restriction enzyme. For example, we have seen type 1, type 2, type 3. Now we are going to see different terms. For example, what is meant by isocycomers, neocycomers, isocadamers and star activity. This is very important definitions. You have to remember. This they can ask in match the following. Okay. So isocycomers means what? That means a pair of restriction enzyme. A pair of restriction enzyme that recognize the same sequence and it will cleave at the same site. That is known as isocycomers. Clear? That means a two different restriction enzyme that will recognize the same site and it will cleave at the same site. That is known as isocycomers. Neocycomers means what? Same restriction enzyme that will recognize the same site but cleaving at different site. Clear? This is known as neocycomers. Then what is meant by isocadamers? Isocadamers means different recognition site will be there, but after cleaving it will produce the same ends. Okay, these examples are also very important. Okay, you just have to remember what is isocycomers, neocycomers, isocadamers and what are the example. Now we are going to see about star activity. Okay, what is mean by star activity? See, for this restriction enzyme activity, we should need an optimum temperature, optimum magnesium concentration, everything should be like optimum condition should be there. If there is an excess of optimum condition, for example, non-optimal conditions are there, that will alter the specificity of the restriction enzyme. Because of that, it will recognize some site and it will cut at the different site. This is known as star activity. This definition they may ask. That's why I'm saying. Okay, that is known as star activity. Okay. So far we have seen what is mean by different class, what is mean by gene cloning, what is mean by genetic engineering, and what are the different requirements needed. What are the different DNA modifying enzymes? Okay. Restriction enzyme, how we are naming the restriction enzyme, examples of restriction enzyme. Any doubt is there so far? Any doubt is there? If no doubt is there means I will proceed. Kindly let me know any doubt is there. Kindly let me know any doubt is there. You guys are listening or not? Any doubt is there? Reply, any doubt is there? Yes, if no other doubt, yeah, clear. Please take marathon class for SRF Biotechnology. Okay, we will start that also. Any doubt is there now? Still no, any doubt is there? No, yes. So we have completed now. Okay, we have completed the different restriction enzyme, everything we have completed. See, just imagine now by using the restriction enzyme, this is my gene of interest. Okay, I have isolated the gene of interest. Okay, now this gene of interest has to be cloned in a vector. Okay, this has to be cloned in vector. This cloned thing I will transform to bacterial species. So now we are going to see what is meant by vector, what are the key characteristics of vector, okay, why we have to use vector, what are the different classification of vectors, everything we are going to see now. Okay. Now you see, first we have to know what is meant by vectors. Okay. Vector is nothing, it's an cloning vehicle. We can say it's a cloning vehicle. Okay. It's a cloning vehicle or a carrier of our gene of interest to the bacterium species. Clear? It will act as a cloning vehicle for carrying our gene of interest to the host organism. Okay. This vector usually we can use, usually plasmid, that means bacterial plasmids can be used as a vector. Apart from that, lambda phase vectors are the different classification of vectors are there that we are going to see now. Okay. Now before going to that, we are going to see what are the key characteristics of vector. If a vector means what are the desirable traits should be there. If you see, the first and most important thing is it should replicate autonomously. Okay, it should replicate autonomously. The size of the vector should be small and easy to isolate and purify. This is JRF 2022 assessment recent question. Okay. The size of the vector should be small and easy to isolate and purify. If the vector size is large, what's the disadvantage? Means while isolation, shearing may happen, that may degrade this plasmids, or another most important thing, if the size of the vector is more, the gene of interest insertion will be less. Clear? So if a small vector means we can clone a large segment of DNA in this. Okay, so this is very important point. Okay, then another important thing is what? It should easy to introduce into the host cell. Okay, it should be easy to introduce into the host cell. Then selectable or suitable markers should be there. Suitable selected markers should be there. Okay, these are the desirable characteristics of a good vector. And most important thing, it should also have high copy number. It should have also have high copy number. Okay, we are going to see one by one. Okay, the first developed vector. Okay, this is JRF 2020 question. Okay. First developed plasmid vector PBR32. PBR32. Okay. It's a plasmid based vector. What question they have asked is, what is mean by BR? We know this P is plasmid. Plasmid BR322. B and R is scientist name. Okay. They have asked this one. Bolivar and Rodriguez. Okay, this question they have asked. Okay, Bolivar and Rodriguez has developed this vector PBR322. The most important characteristics of this vector is it will have two selection marker genes. One is ampicillin resistant gene and second one is tetracycline resistant gene. This is clear. So two resistant genes will be there. One is ampicillin resistant, second is tetracycline resistant. If our gene of interest is inserted either of this ampicillin or in tetracycline region, okay, the function of this resistant gene will be depressed. Because of this bacterium will lose the ability to grow at particular antibiotic. Okay. Based on this, they have selected whatever the colony is having our gene of interest. Okay. This is selection procedure. Clear? If my gene of interest is inserted in ampicillin resistant gene region, so this E. coli carrying the particular plasmid will lack the resistance to this ampicillin. Okay. So I can make a replica plate technique. Based on this, I can select the clones that is having my gene of interest. I hope this is clear for everyone. This we have clearly discussed in our lecture series. Okay. Then second and most important thing, most important vector is plasmid University of California 8 vector PUC. This full form they have asked. Okay. University of California vector. Here the most important thing is they have asked which type of selection procedure they will use. Okay. Here they will use blue white screening. Blue white screening. This they have asked 3 times. JRF 2021, 2019 and 2023. 3 times they have asked. Okay. Blue white screening. In PBR 322 we have seen if our gene of interest is inserted to the antibiotic resistant gene, the resistance toward that particular antibiotic will be decreased. Clear? But in plasmid University of California, the gene of interest will be inserted in laxed region. Okay. Laxed region. Okay. If my gene of insert is inserted here, the laxed function will be disturbed. Okay. So my recombinant will become white colonies. This is the question they have asked. Recombinant will have white colonies and non-recombinant will produce blue colonies. Okay, this is the question they have asked. If you see here, okay, this is the colonies produced. You see here, blue colonies are there and white colonies also there. Blue colonies also there, white colonies are there. This white colonies are what? These are the recombinant. Due to insertion of the gene of interest, the laxed function was disturbed. Because of that, it producing white colonies. So, recombinant will be white colonies. Clear? Recombinant will be white colonies. Okay. Non-recombinants will be blue colonies. It will produce blue colonies. Clear? This is very important question. Okay. They will ask this one only. Okay. Another important thing. We can use lambda phage vectors also. Okay. The most important thing, what question they will ask about lambda phage is the size of the lambda phage genome is 49 kb, approximately 49 kb will be there. Approximately 49 kb will be there. And gene of insert cloning size, how much segment of DNA we will clone in lambda phage vector? This they will ask. Okay. This lambda phage vector classified into two types. Okay. Insertional vector and replacement vector. Two types are there. Okay. You see here clearly, insertional vector means our gene of insert we will insert within the gene of this lambda genome. Okay. So insertional vector, the cloning capacity will be always less. This is the statement question they have asked. Insertional vector will have cloning capacity of only 7 to 10 kb. Okay. But if you see in replacement vector, some of the stuffer fragment we will remove from the lambda genome. Because of that, the insertion fragment will be more. 11 to 20 kb of gene of interest can be inserted here. Clear? So, insertion vector means around 7 to 10 kb of DNA we can clone, but replacement vector means up to 20 kb of gene can be cloned in the replacement vector. This is the key point they will ask. Okay, this is the most important thing. Okay, another most important thing. Okay, page M13 vectors. Page M13 vector, the specialty we are going to see about exam point of view only. Page M13 vector means the genome of the page M13 is single stranded. So this will be primarily used for synthesizing the single stranded DNA molecule. Okay, this can be used in sequencing. Okay, this is the objective point they can ask. Okay, so M13 vectors are specifically used for sequencing purpose. Okay, synthesizing of single stranded linear DNA genome for sequencing purpose. This is the most important thing. Okay, apart from that, different things are the cosmid vectors. Key point, cosmid vector means that will have a cloning site, that means have a cloning size of up to 45 kb. This is the key point you have to remember. Then why we are saying cosmid? If you separate this into two words, it will have lambda page cos site plus plasmid will be there. Okay, that's why we are saying this as cosmid vector. Okay, the cosmid vector will have a cloning capacity up to 45 kb. We are going to see the key points only. Okay, another most important thing, bacterial artificial chromosome and yeast artificial chromosome. Clear? Bacterial artificial chromosome means that will have a cloning capacity up to 300 kb. Okay, 300 kb of cloning capacity will be there in bacterial artificial chromosome. Another thing is yeast artificial chromosome. YAC, yeast artificial chromosome. This was developed by David Burke. Okay, David Burke only developed this yeast artificial chromosome. If you see the DNA cloning capacity, we can clone up to 1000 to 2000 kb. Clear? We can clone 1000 to 2000 kb of DNA. Okay, 1000 to 2000 kb of DNA segment can be cloned. Then another most important thing, they have asked this SUP4. This is the selectable marker used for screening the recombinant in YAC vectors. Okay, clear? SUP4 is a selectable marker used for selecting the recombinant in YAC vector. These are the key points. This they have asked in JRF 2024. Okay, that's why I have highlighted here. Okay. These are the yeast cloning vector. There are different types of cloning vectors are there. Okay. Integrative vector, episomal vector, centromeric vector and YAC vector. These are just classification you have to remember. Okay. These are not that much important. The most important thing is yeast artificial chromosome. What is the insert size? Okay. This is just a brief exam point of view. You see here, if PBr322, that will have a cloning capacity of up to 10 kb. Okay. Lambda phage vector, it will have a cloning capacity up to 25 kb. Cosmid means what? Up to 45 kb. Phosmid means 40 kb. BAC, HAC, this we have seen. 300 kb will be there in bacterial artificial chromosome. Yeast artificial chromosome means up to 3000 kb will be there. Human artificial chromosome, this is more advanced one. It will have up to 2000 kb. Okay. The question what they will ask from this vector means, what are the key characteristics of vector? What are the different selection procedures? And what are the DNA insert size for each and every classification of vector? Any doubt is there? Kindly let me know, any doubt is there? Any doubts? Any doubts? No doubts. Yes. Yes, yes, very good. See, now we have seen what are the different classification of vector. Okay, we have seen what is bacteria artificial chromosome, yeast artificial chromosome, everything we have seen. Apart from the three different things are there. Okay, this you have to know only the definitions. Okay, this is very important. What is meant by shuttle vector, what is meant by binary vector, what is meant by co-integrated vector. These three definitions are very important. Okay, they have asked this several times. Co-integrated vector, shuttle vector, and binary vector. These are the three important types of vector you have to know apart from this. Okay. Before going to that, we will see a brief introduction about TA plasmid and RA plasmid. We know this TA plasmid, tumor inducing plasmid, RA plasmid, this will present in agrobacterium. Okay, agrobacterium it will have. The key question what they will ask from this is, what is the size of this TA plasmid? It will be approximately 200 kb will be there. This is the most important thing you have to remember. The size of the TA plasmid is approximately 200 kb, but the tDNA region size will be approximately 25 to 30 kb. Clear? In your question, what they are asking, you have to see whether they are asking about TA plasmid or whether they are asking about tDNA region. Okay, based on this, we have to answer. Okay, if TA plasmid means size will be 200 kb, if tDNA region size means 25 to 30 kb. We are going to see about what is meant by shuttle vector. This they have asked in 2024. Okay, shuttle vector means nothing. Okay, it will have two different origin of replications. Two different origin of replication will be there. Okay. You see, a cloning vector that replicate in two different host organism is known as shuttle vector. Okay. This is the definition they have asked. A cloning vector that replicate in two different organisms like E. coli and Saccharomyces cerevisiae yeast is known as shuttle vector. Okay. Now we are going to see what is binary vector. Binary vector means simple. Okay. Here, two different vectors they will use. One vector will have particularly of virgins. Okay, virgins will be there. The second vector will have all the things, the gene of interest, everything will be present in the second vector. We are going to see this, why the virgins will be present separately in one vector. Okay, this virgins will release the vir products. These vir products will help in integration of the tDNA into host organism genome. Clear? This we are going to see briefly in upcoming slides. Okay, so binary vector means a plant transformation vector system in which tDNA and virulence genes are present on separate plasmid. Clear? tDNA and virulence plasmid are present on separate plasmid. Another thing, what is meant by co-integrated vector? Co-integrated vector means what? Here the same thing, two different vectors will be there, but homologous recombination will happen between these two vectors. Because of this, the tDNA will be inserted. Clear? So, homologous recombination will be happen here. See here the definition. Co-integrated vector is a recombinant plasmid created by fusing smaller plasmid carrying gene of interest with a larger disarmed term plasmid. Okay, clear? I hope this is clear for everyone. Okay, the definition only you have to remember properly. Okay, the definition only you have to remember properly. So far, we have seen different class of enzymes we have seen, restriction enzymes we have seen. Okay, what is vectors we have seen. Okay, if you see, this is the genomic DNA. From this genomic DNA, I have isolated the gene of interest. This is my gene of interest. This gene of interest has to be cloned in vector. This is the vector I have seen. Okay, we have discussed about the vectors. Okay, if this gene of interest has to be ligated in the vector, first the vector has to be cut with the restriction enzyme. I have cut it with restriction enzyme. So this gene of interest has to be ligated here. So DNA ligase enzyme I will use. Here comes the another key thing. What is meant by adapters and what is meant by ligators? Adapters and ligator. If the vector is producing sticky ends and the gene of interest also will have sticky ends, this will go and bind here without any help. Clear? Then DNA ligase will join that one. But if it is producing blunt end, okay, how to convert this blunt end into sticky ends? There comes the concept of adapter and ligators. Okay, simple concept. You see, adapter means what? Means it will have a preformed cohesive sites will be there. Okay, see, this is the adapter. It will have preformed cohesive site. So what they will do means this adapter at excess concentration will be mixed with the blunt ended DNA. So you see here at last the adapter was ligated with our blunt end DNA. What is meant by linkers? Linkers means it will have recognition site for different restriction enzyme. Okay, see here, this is the blunt end DNA I have isolated. These are the linkers. I have joined the linker. Then I will treat with the specific restriction enzyme that will convert the blunt end into sticky. And I hope this is clear. Adapter and linker. This is repeated question. What is mean by adapter? What is mean by linker? Okay. Now we have our complete vector. Vector having our gene of interest. Okay. Vector having our gene of interest. Now we have to transfer this into the host bacterium. Clear? Now we have to transfer this to our host bacterium. What are the different ways to transfer this? Our vector having gene of interest to host bacterium, that we are going to see now. Okay. This is most important definition. Okay. What is meant by transformation? Okay. Transformation means what? The process by which bacterial cell take up the naked DNA molecule is known as transformation. This is very, very important definition. Transformation means what? The process by which bacterial cell take up the naked DNA molecule is known as transformation. Okay. There are two different types of transformations are there. Chemical transformation and electroporation. Okay. Chemical transformation means what? First, they will make the bacterium cell as competent. Competent means what? The ability to uptake the DNA from external environment. Okay. They will loosen the cell wall. They will loosen the membranes of the bacterium. Okay. That we will known as competent cells. Okay. Bacterial cell capable of taking the DNA are called as competent cell. How they will make this? How they will make this? This is another important question. Okay. They will do repeated heating and cooling. Okay. Heat shock they will give at 42 degrees Celsius. Then they will transfer at ice. Okay. Because of this, the cell become competent. Okay. The most important question what they will use ask means calcium chloride will be used. Okay. Calcium chloride chemical will be used. Okay. While making the competent cells. This is the most important thing. Okay. You see here, this is the normal bacterium. We will use calcium chloride treatment. The cell become competent. Our plasmid containing the gene of interest will be bind here. After that, it will enter inside the bacterium. Clear? So, the gene of interest along with the vector has came inside the bacterium. Okay. Now, another technique is what? Electroporation. Here, they will take the bacterium and they will treat with heat shocks. Okay, then electric pulse they will give. Because of this electric pulse, what will happen means temporary pores will be formed on the membrane of the bacterium. In that pores, the DNA molecule will be entered inside. Clear? This is just a simple thing. Electroporation and chemical method. Okay, these are the two different methods. Okay, two different methods. Still now we know the bacterium will having our gene of interest along with the vector is present here. Okay. There may be three different possibilities are there. These are the three different possibilities. Okay. This bacterium will have our gene of interest along with the vector also will be there. Okay. Without ligating the gene of interest, some of the self-ligated vectors also will be there. Okay. In some of the cases, the gene of interest may be inserted in wrong position. That also will be there. Now we have to select only the bacterium having our gene of interest. Clear? How to select the bacterium only having our gene of interest? While our vector class itself I have told, there are different selection strategies are there. Blue-white screening is there first thing. Blue colonies are non-recombinant, white colonies are recombinant. Then antibiotic selection are there. That is the things. Okay. So blue-white screening will be there. Apart from that, lag-Z screening will be there. There comes the role of selectable markers and screenable markers. Okay, this we are classified as selectable markers and screenable markers. The most important thing, they will ask the example for selectable markers and screenable markers. Okay, selectable marker, these are the example of selectable markers. Okay, mostly if you see the selectable markers are antibiotic resistance genes. Okay, you have to read in this slide what are the different selectable markers and what is the function, how the antibiotic resistance is providing to that bacterium. For example, I am taking these things, neomycin phosphotransferase. What that gene will do? It will phosphorylate the neomycin compound. Because of that, that will become inactive. Clear? So, if the neomycin phosphotransferase gene is present in the bacterium, the E. coli will be resistant to that particular antibiotic. I hope this is clear. For like that, you have to read for this. Okay, what are the bacterium? What are the antibiotics? What is the mode of action? Whether it's an example of selectable marker or scorable marker. Clear? Now we are going to see about scorable marker, selectable marker and scorable marker. Selectable marker means what? That will be majorly an antibiotic genes. Okay. In scorable markers, that will produce a visual products. Okay. For example, you see here chloramphenicol acetyltransferase, beta glucuronidase, firefly luciferase, green fluorescent protein. These are the typical example of scorable markers. Okay. Then most important thing, scorable marker is also known as visual marker or screenable marker. You shouldn't be confused with selectable markers. Okay. I am writing here for your clarity. Okay. Selectable marker. Selectable marker. Okay. This will be mostly antibiotic genes. Okay. Antibiotic resistant genes. Okay. Another class is there, scorable, screenable or visualable. These all are same only. Okay. From here, sure they will ask one question. Okay. You shouldn't be get confused. From here, sure they will ask one question. Okay. This we have already discussed. There are two different methods are there. One is what? Antibiotic resistant selection is there. Second thing is what? Blue-white screening is there. White colonies means it will produce, it's a recombinant. Blue colonies means it's a non-recombinant. This we have already discussed. Okay, we have seen what is meant by transformation. Okay, transformation means uptake of the DNA by the host bacterium. If that DNA is from phage, bacteriophage, the same process only, but the DNA is from phage DNA, we can say that as transfection. Okay, transfection, the simple definition. Okay, if it's a phage DNA, we will say that as transfection. This definition also they have asked in 2023. They asked this one. What is mean by transfection? Those who have attended our PYQ sessions, they have know this one. Okay, what are the question they have asked from this syllabus? Okay, okay. Then same thing, how the selection will be there based on lag-Z screening or lambda CL screening. This we have already discussed in our lecture class itself. Okay. Any doubts are there so far? Any doubts are there? Any doubts? Any doubts are there? Kindly reply. Are you guys are watching or any doubt is there? Now we have completed cloning completely. What is mean by cloning? What are the enzymes, vectors, everything? Yes. Yes, no doubts. Okay, we can have break for 5 minutes. Okay, we will resume at 6:23. Okay, we can have break for 5 minutes. Huh? 6:23, I will see you all. Okay, 6:23. Shall we proceed? Yes, kindly reply, shall we proceed? Any doubts so far? Shall we proceed? Yes. Yes, we have so far we have seen how a gene of interest can, yeah. So far we have seen how a gene of interest will be cloned. Okay. Now we are going to see some of the minor concepts. Okay. What is meant by cDNA cloning and what is meant by genomic DNA cloning. Okay. The name itself is there. Okay. cDNA cloning means the complementary DNA, that are the expressed messenger RNA, will be converted into DNA, that is cDNA. This will be cloned in vector. Okay, that is known as cDNA cloning. Okay. Genomic DNA means the genomic DNA will be isolated, that will be converted into different fragments, then that will be cloned in the vector in bacterium. Okay. The most important thing what they will ask from this cDNA and genomic cloning is, see, cDNA clones will always contain only the expressed genes. Clear? cDNA will have only the expressed genes, but the genomic DNA will have both exons as well as intron sequence. This is the most important question they will ask. And most important thing, another thing is how we can convert the messenger RNA into cDNA. Okay, how we are converting messenger RNA into DNA molecule. Okay, this is most important. There are two different ways are there. Okay, first is self-priming method. Self-priming method for synthesis of cDNA. Second one is replacement method. Okay, replacement method. We know the messenger RNA will have 5' cap structure and 3' poly A tail will be there. This we know. If 3' poly A tail is there, we can use oligo-DT primers. Oligo-DT primers, then we can add our DNTPs along with the reverse transcriptase enzyme that will lead to synthesis of the first strand. This is the first strand of the cDNA. Clear? This is the first strand of cDNA synthesis. Okay. The second strand of cDNA will be synthesized by these two methods. One is self-priming and second one is replacement method. Okay. In self-priming, if you see, what they will do means, they will treat this DNA RNA hybrid with alkaline treatment. Okay, because of this alkaline treatment, we know the RNA molecules are susceptible to alkaline treatment due to the presence of 2' hydroxy group. This also they have asked one time, 2024 JRF question. Okay, so the RNA segment will be degraded. Only the first strand synthesis will be exist. Now we are going to see about self-priming. Here what it will do means, here an hairpin loop will be formed in the DNA, first strand of the DNA. This will act as a primer for synthesizing of the second strand. I hope this is clear for everyone. Okay. The most important thing what you have to remember is, in self-priming method, there will be a hairpin loop will be formed that will act as a primer for synthesizing the second strand of cDNA. Okay. But if you see in replacement method, the first strand will be there, then they will treat with RNSH. Because of this, the RNA molecule will form a nix. Different nix will be there. This nix will be act as a primer. Then here the DNA polymerase enzyme will come that will synthesize the second strand. Okay, clear? So replacement synthesis and self-priming methods are two different method of DNA synthesis. Okay, cDNA synthesis. Okay, if you want to practice topic-wise subjective questions, okay, kindly enroll in our test series. And those who are targeting for ICAR 2027, okay, kindly enroll in our course. It will be very much useful for your preparation. Okay, I will assure you it will be very much useful for your preparation. Okay. Now we have seen what are the different methods of cDNA synthesis. Okay. Now how to clone the synthesized cDNA into vector. Key points only we are going to see. Okay, this is our cDNA. Okay, this cDNA, this will be cloned into the vector. Here another important thing, for cloning of cDNA, low capacity vectors can be used. For example, plasmid vectors can be used. But for cloning of genomic DNA, we have to use high capacity vector because the genomic DNA size will be more. So that vector capacity also should be more to carry the gene of interest. Clear? So for cDNA cloning, even plasmid vectors are used. But for genomic DNA cloning, lambda phage vectors are highly preferred. Apart from that, what? Bacterial artificial chromosome and YAC will be preferred. Okay, this is the key difference between cDNA cloning and genomic DNA cloning. Okay. Now, we have cloned our gene of interest, that is cDNA or genomic DNA, to the vector. What are the different selection strategies for selecting the specific clones? Okay, these are the different selection strategies. You just go through once. Okay, nucleic acid hybridization, colony hybridization, plaque hybridization, PCR screening, immunological screening, and functional complementation. These are the different techniques that are used for specific clones. Okay, we will see the key points about each and every techniques tomorrow. Okay, tomorrow we have our technique class. In that I will cover all the things. Okay, don't worry. So far we have completed what is meant by cloning, how we can clone the cDNA, and how we can clone the genomic DNA into the vector. Okay, clear? This is the basic about the DNA, like gene clonings and recombinant DNA technology. Okay. Now we are going to see our next topic. Okay. Plant transformation. If we want to transfer in plants, what are the different techniques are there? Okay. From here, sure they will ask one or two questions. So listen properly. What are the different plant transformation method? Okay. For example, we want to produce vitamin A rich rice, that is beta carotene rich rice. Some of the gene, external source gene, has to be inserted to the plant. Correct. So how we are inserting that external gene into the plant? That is what transformation. How we are transforming the genes. Okay, different techniques are there. We can broadly say vector mediated gene transfer and vectorless mediated gene transfer. Okay. So vector mediated gene transfer, or that can also called as indirect method of gene transfer. Okay, vector is known as what? Indirect method of gene transfer. Okay. Then second classification is direct or vectorless gene transfer. Okay. Here subclassification was there. Okay. Physical method, chemical method, and DNA imbibition. Okay. This example are very important. Okay. Examples are very important. Okay. The physical method, if you see electroporation, electric pulse we can give. This is JRF 2020 question. Okay. Biolistic is also known as particle bombardment method of gene transfer. It's an example of physical method of plant transformation method. This is the question they asked. Okay. Biolistic method of gene transfer is also known as particle bombardment method. Okay. This is an example of physical method of gene transfer. Okay. Apart from that, what are there? Microprojection, liposome encapsulation, and silica carbide fibers, that is nanofibers we can use. These are the physical methods. Okay. Chemical methods means what? We can use polyethylene glycols, DEAE we can use, or calcium phosphate precipitation methods we can use. Okay. Then coming to the biological method of gene transfer. In biological, or we can say vector method or indirect method. Okay. Here we can use bacterium or virus, okay, as a source, okay, as a source for transferring our gene of interest. If you see, agrobacterium mediated gene transfer is there, or plant viruses we can use as an tool for transferring our gene of interest to the plant genome. Okay, I hope this is clear for everyone. Now we are going to see A to Z of agrobacterium mediated gene transfer. This is most important topic. Agrobacterium mediated gene transfer. Okay. If you want to do CRISPR-Cas genome editing, whatever genome editing technique you are using, The gene transfer is one of the key bottleneck. Primarily, they will use agrobacter mediated gene transfer method. So this is very important point. You have to listen properly. Okay. First, we will see the basic of Agrobacterium tumefaciens. Okay. Agrobacterium tumefaciens is a soil bacterium. It's a gram-negative bacterium. This we know. Okay. Here, two different strains are there: Agrobacterium tumefaciens, Agrobacterium rhizogenesis. Okay. Tumefaciens will have tumor-inducing gene. Rhizogenesis will have RI plasmid will be there. Okay, RI plasmid will be there. What this tumor inducing plasmid will do? Okay, this is the question we have to answer. Okay, if you see this TA plasmid, this will assist in integrating the tDNA into the host genome. Okay, the tDNA segment of the TA plasmid will be integrated into the genome, plant genome. Because of that, crown gall disease will form. Okay, so if we replace the tDNA with our gene of interest, that can go and integrate in the plant genome. This is the concept they have developed. Clear? So whatever gene we have to integrate it in plant, that will be inserted in the tDNA region. So that will be go and integrated with the plant genome. Clear? Now we are going to see the basics of this TA plasmid. This we have already discussed. The size of the TA plasmid will be around 200 kb. The size of the tDNA region will be around approximately 25 to 30 kb. Okay. JRF 2025 question what they have asked means what are the genes that are present in tDNA regions? Okay. If you see here clearly, the tDNA region will have genes for oxygen biosynthesis, cytokinin biosynthesis. Apart from that, opine biosynthesis and catabolytic genes will be there. Okay. In the option what they have given means four options was there. Okay. Four options was there. In all the option they in except one option they have given virulence genes also will be present. Virulence gene also will be present in tDNA region, like that they have given. Virulence gene will never present in the tDNA region. Clear? So that will be present separately in the TA plasmid. So this is the question they have asked. Okay. This we have already discussed. 200 kb size will be there. 15 to 30 kb the tDNA region size will be there. That will be flanked by, if you see here, this tDNA region is flanked by right border and left border sequence that they have given. Right border and left border sequence will be there. Okay. Now we are going to see the mechanism, actual mechanism, how this tDNA is integrated into the in the plant genome. Okay. How the tDNA is actually integrated in plant genome. Okay. For this integration, different Vir genes will function. Okay. I already shown in the figure. Okay. tDNA region will be there. This tDNA will be integrated in the plant genome. For assessment of the integration, different Vir proteins will come. This is the question they are going to ask. Okay. What are the different Vir genes and what is its function? Okay. If you see this Vir genes will encode a proteins. Okay. This Vir genes will encode proteins. This protein will assist in integration the tDNA in the vector. Okay, tDNA in the plant genome. Okay. If you see here, there are 8 different operons will be there. 8 different Vir operons will be there. Among them, Vir A and Vir G are Vir A and Vir G are most important. Okay, Vir A and Vir G are most important one. Why it is more important? We are going to discuss now. Okay, see, this is the basic. If a plant is there, that will release the phenolic compounds. Okay, particularly astosyringin can be act as a signaling molecule. If the astosyringin is there, for example, signaling molecule is there, this will be pursued by Vir A. Okay. So, Vir A is the phenolic sensor. Phenolic sensor is what? Vir A. If Vir A is sense this phenolic plant phenolics, this will become active. then it will phosphorylate Virg. Okay, Virg will be phosphorylated. This will go and activate other virgins. Clear? This will go and activate other virgins. Okay. You just see here, this is the tumor inducing plasmid, TA plasmid. This is the tDNA region. See, different Vir proteins are there. Okay. The most important Vir protein is Vir D2 and Vir E2. Okay. This Vir D2 and Vir E2, what it will do means, this Vir D1 will be there. This will act as an endonuclease. So this tDNA fragment will cut and come here. Where D2 will bind with the tDNA to prevent from the nuclease activity. Okay. This is a single stranded molecule. So single strand binding protein will come and bind here to maintain its stability. This single strand binding protein is Vir E2. Clear? So this has to be integrated in plant genome. How it will go and integrate? You see here, the bacterium will come and attach on the surface of the plant cell. It will form a sex pilus-like structure for transferring the tDNA into host plant genome. This will be formed by Vir B. Okay, Vir B will be assisting in formation of this sex pilus-like structure. Through this pilus-like pore, thus the tDNA has came to the plant. Okay, this will go and integrate in the genome of the plant. This is the different Vir gene function you see here. Vir A, what it will do? This is the key question they are going to ask. Okay, listen properly. If you have any doubt, kindly let me know. So, Vir A is what? It's a phenolic sensor. It is the first Vir protein that will sense the phenolic compound. Okay. Second thing is what? This Vir A will autophosphorylate this Vir G. This will activate all other Vir proteins. Okay. We have already seen this D1 is what? It's an endonuclease that help to nick the border sequence of tDNA. Okay. Once the single stranded tDNA is formed, this Vir D2 will come and bind here to prevent from nuclease activity. So, the cut DNA border remained attached to 5' end of the tDNA and guides the transfer to the nucleus. Okay. Then what this Vir E2 will do? It will act as a single strand binding protein. It will come and bind here. This is the key important Vir genes. Apart from that, what are the things will be there? Vir B, this will form a type 4 secretion system. That means what? Sex pilus-like structure for transforming the tDNA into host organism. Clear? Any doubt is there? The mechanism for tDNA gene transfer is clear. Any doubt is there? Any doubts? Yes, any doubts? If no doubts means I will proceed. Kindly let me know any doubts is there. No doubts, yes. Yes, we can proceed now. Okay. Now we have seen how the tDNA is integrated into the host plant. Okay. This technique we can use for integrating some of the genes to the plants. In spite of tDNA, we can insert our gene of interest to the particular region so that gene can be go and insert to the plant genome. Okay. Now we are going to see our next topic of today's session. Okay. Transgenics in crop improvement. From here, again I am saying today I have taken the most important sections only. 3 to 4 questions they can ask out of 30 questions. We know around 100 question is there from subject, biotech, physiology and biochemistry. Among them 3 to 4 questions will come from this topic, transgenic in crop improvement. Okay. Transgenic in crop improvement. So listen properly. If you have any doubt, kindly let me know. Okay. So why we have to read about the transgenics in crop improvement? For example, we have already seen if some of the trait we have to incorporate in the existing genotype. For example, rice plant is there. We have to enhance the beta carotene content in that rice grains. What we can do, the beta carotene biosynthetic genes can be incorporated in the rice genome. Because of that, the beta carotene content can be increased. Okay. Then different things are there. If you want to develop some of the resistant plants, for example, viral resistant plants, disease resistant, insect resistant plant, the transgene has to be inserted to the genome of the plant. Okay. So how they are doing this? What is the importance of the transgenics in crop improvement? That we are going to see now. Okay. So we are going to see in different aspect. Transgenic for abiotic resistant, biotic resistant, quality improvement, herbicide tolerance, male sterility, like that different aspects are there. Okay. Before going to start that session, we have to know what are the key questions they can ask from this. Okay. The most important thing you have to remember is what are the different transgene? What are the different transgene they are using for developing particular trait? Okay, for example, cry genes will be there, crystal proteins will be there. This can be used for developing insect resistance. Like that, different transgenes will be there. Okay. Then second most important thing, what are the commercialized variety for each and every crop along with the traits? For example, in 2023, they have asked Freedom 2 is an variety of which crop for which trait. Okay. It's a variety of papaya for virus resistant. Like that also they can ask question. Okay. So you have to know what are the different transgene, what is the source of the transgene, and what are the commercialized varieties. Okay. Now we are going to see transgenic for biotic stress resistance. Okay. Transgenic for biotic stress resistance. If you know, biotic resistance means what? Insect will be there, viral will be there, or fungal and bacterial disease will be there. Okay. Just we are going to brush up the knowledge. Okay. You see, transgene, the most important thing for insect resistant is what? BT gene. Okay, BT gene from Bacillus thuringiens. Okay, who has discovered this? Ishvatari in 1901 from disease silkworm. Whatever I am underlining, these are the key points. Okay, so this Bacillus thuringiens will have the BT genes that will synthesis cry proteins, that is crystal proteins. Okay, these crystal proteins are active under alkaline condition of the larval stomach. What it will do means it will make pores in the intestine or pores in the membrane of the larva. Because of that, lysis will happen, the larva will die. So what we can do, we can express this BT gene. Okay, in cotton we know BT cottons are there. So if the insect will feed that one, this BT gene will produce cry proteins. This cry proteins will make pores on the membrane. Because of that, lysis will happen, the larva will die. So this is a simple idea. Okay, so what question they may ask? They have already asked about the Bacillus thuringiensis, who has discovered everything. So this year I am expecting, if they want to ask some deeper question, means there are different classification of cry proteins are there. Okay, cry 1, cry 2, cry 3, cry 4, cry 4, like that different classification are there. So they may ask which cry proteins will be used for targeting which class of pest. For example, cry 1 and cry 2 will be particularly used for targeting the Lepidoptera pest. Okay, Lepidoptera pest. Cry 3, if you see, cry 3 is specific for Coleoptera. Okay, you just remember A, B, C, D. Okay, C is third letter, so cry 3 is for Coleoptera. Okay, cry 4, D, D for Diptera. This is most important. Okay, cry 3 for Coleoptera, cry 4 for Diptera, cry 5 for Lepidoptera and Coleoptera. This question they may ask. Okay, what are the different cry proteins with their target insects? Okay, then we are going to see about other from BT, what are the different transgene they can use. Okay, BT they can use. Apart from that, alpha amylase inhibitor, protease inhibitor, these are the different genes. If we express these genes, we can produce a insect resistant plants. This is the concept. Okay, insect resistant plants we can produce. Okay, This is very important slide. Okay, these are the different cry proteins they have expressed in different crops along with the commercialized plant product. Okay, if you see, Bolgard is a cotton crop with expressed with cry1 AC protein. Okay, Bolgard, yield guard in maize, maximizer also in maize, new leaf in potato. Okay, new leaf for potato along with the trade. This is the question they are going to ask. Okay, what are the different cry proteins expressed for which crop and what is the product name developed? Okay, so this is the key question they will ask. Okay, then another thing for disease resistant and for virus resistant. Okay, for virus resistant, what they can use? They can use code proteins, satellite RNA. Virus resistant is not that much important. Just you go through once. These are the different genes. But what are the varieties they have developed is very much important. Okay, varieties developed was very much important. I already told this they have asked in 2024. Okay, Rainbow and Sunnyemp, Freedom 2, Destiny and Liberator. This all are viral resistant crop. Okay, viral resistant crop developed. Okay, viral resistant crop. Okay, coming to the disease resistant, if you want to make a plant resistant to disease like fungal disease or bacterial diseases, the key important thing you just remember, the fungus cell wall will have what? Chitins will be present in cell walls will be there, then glucans will be there. If we will degrade that chitins and glucans, okay, resistance can be enhanced. Clear? So we can use chitinase gene, beta-1,3 glucanase. Then apart from that, we can use pathogenesis resistant proteins, stilbene synthase genes. These are the specific classes of the genes. If we overexpress in the plant will become disease resistant. Okay, the plant will become disease resistant. Okay, so far we have completed the biotic stress. How we can develop a resistant against insect, pest, virus and disease. Now we are going to discuss about abiotic stress. Okay, this is another important thing, abiotic stress. Here, what question they will ask? Okay, in abiotic stress, they will repeatedly they are asking about fatty acids only. If you just listen here, if heat stress, you have to increase the heat stress tolerance. If you want to increase the heat stress tolerance, we have to enhance the accumulation of saturated fatty acids. Okay, saturated fatty acids. If you want to enhance the cold stress tolerance, we have to enhance the accumulation of unsaturated fatty acid. This is clear. This is most important thing. Okay, heat stress should be enhanced means more amount of saturated fatty acid should be there, and cold stress should be enhanced means more amount of unsaturated fatty acid should be expressed. I hope this is clear for everyone. Okay. Okay. Another important thing, herbicide resistant. This they have asked in 2022. Match the following. Okay, 2022 match the following question they have asked. Okay, 2022 match the following. What question they have asked means they have given the different resistant genes. Okay, different resistant genes they have given, and what is the target herbicide? Okay, what is the different herbicide gene? Along with that, what is the its herbicide tolerant will be produced. Okay, bar gene will be resistant against glufosinate. Okay, ROA and GOX, this will be resistance against glyphosate. This you just go through once. Okay, this is very important slide. Just go through once. Okay, ALS, bromoxylin. Okay, what is the key herbicide resistant gene? Okay, what is the target herbicide and which enzyme it will inhibit? This also you go through once. Okay, this question they asked in 2019. Okay, this they asked in 2019. What is the mode of action? How the bromoxylin will be converted into detoxify product? Okay, bromoxylin will be converted into 3,5-dibromo 4-hydroxybenzoic acid. This is the degraded product of bromoxylene. Any doubt is there in this? Any doubt? You just see that any doubt is there, kindly let me know. No doubts. No doubts, yes. No, yes. So far we have completed herbicide resistant, abiotic and biotic stress resistant. Okay. Now we are going, see, before going to that, we can see some of the examples. Okay. What are the commercialized crop? Okay. I am just giving one shortcut to you. Okay. Wherever Roundup is there, Roundup ready. Okay. Wherever Roundup is there, that will be specific for glyphosate resistant. Clear? This is the question they will ask. Okay. Roundup ready is specific for glyphosate resistant crop. Clear? Glyphosate resistant crop. Okay. That is the question they will ask. Okay. Now we are going to see transgenic for quality improvement. Transgenic for quality improvement. Okay. See here, quality improvement. How to increase the shelf life. This we have already discussed. Flavor saver tomato for delayed ripening, antisense RNA technology by Calgene company. Okay. Apart from that, we know this ethylene is the primary hormone involved in fruit ripening. Okay. If we decrease the ethylene content by targeting the biosynthetic genes, the ethylene accumulation will be less. Yes, ethylene accumulation will be less. So they have targeted ACC genes. Okay. ACC genes also they have targeted. Okay. By this we can improve the quality of the produce. Okay. You see here antisense RNA technology for developing flavor saver tomato by Calgene company. They have targeted the polygalactonase gene. This they have asked one time. Which gene they have targeted? Okay. Polygalactonase gene they have targeted. Okay. Polygalactonase gene. Okay. Another thing is what? Overexpression of SAM hydrolase. Okay. What it will do means it will convert ACC2 into other metabolites than ethylene. So, ethylene levels will be less. Because of that, fruit ripening will be delayed. I hope this is clear. Now, we are going to see how to improve the shelf life in the flower. Similar aspect only. Petal senescence, if we want to decrease, means we have to target the genes that are involved in ethylene biosynthesis. So, they have target here antisense AC oxidase. Okay, ACC oxidase gene they have targeted here. Just to go through this. Okay, just to go through this. Okay. Then most important thing, what are the transgenic varieties developed in carnation for which trait? They have asked this one time. Okay. So the transgenic variety developed in carnation is moonlight, moonshade and moonshadow or moon vista. This is the transgenic variety developed in carnation. Okay, carnation flower. What is the trait means? Purple color flowers they have developed. See, purple color flowers they have developed. How? They have integrated this gene, flavonoid 3,5 hydroxylase gene. Okay, flavonoid 3,5 hydroxylase gene. Because of this, delphicidin was accumulated in the flowers that will give purple color. Clear? That will give purple color. Okay. Next, golden rice. Okay. We have seen up to now quality, how to improve the quality. Okay. Now we are going to see nutritional aspect, how to increase the nutrition. For example, golden rice. Okay. Golden rice. 2 is there, golden rice 1 and golden rice 2. Okay. The key important thing what they will ask from golden rice 1 and 2 is what are the genes and what are the sources of the genes they have integrated in the rice genome. Okay. For example, in golden rice 1, they have taken phytoin synthase gene and lycopene cyclase gene from daffodils. This is the key important question. Daffodils, you have to know about the scientific name also. Okay. So they have taken the phytoin synthase and lycopene cyclase gene from daffodils. Okay. Then Phytoin desaturase gene they have taken from Erwinia viridivora. Okay, this is the key important point. Okay, what are the genes they have taken and what is the source? And if you see in golden rice too, they have taken the phytoin synthase gene from maize. Then they have used endosperm specific promoters. Because of that, they have obtained 23 times more beta carotene in seeds comparing to golden rice one. Okay, by changing the source of the gene, they have taken phytoin synthase gene from maize. Because of this, higher beta carotene accumulation was there. I hope this is clear for everyone. Okay, these are the different varieties. You have to just focus on this thing only. Okay, moonlight, moon shadow, moon vista. Carnation, flower color, this we have already seen. Flavor saver, endless summer. This is for improving the shelf life. This we have already discussed clearly. Okay. Now this is the last topic, transgenic for male sterility. Okay. If you want to produce a sterile line, we can express a taptum specific. What is meant by taptum? Taptum is the tissue that will help in transporting the nutrient from plants to the pollens. Okay, from the pollen transfer will be there. If in the taptum specific promoter if we use and if we express some of the toxic genes, the pollen's viability will be decreased. Because of that what? The plant will become sterile. So if a plant should be sterile, we can express this Barnes gene. Okay, Barnes gene with a taptum specific promoter. This is very important. Taptum specific promoters. Okay. So if we use that means this will includes ribonuclease enzyme. Whatever the RNAs will be produced in the pollen, this will be completely degraded. Because of that, the plant will become sterile. The most important question what they will ask means, what is the source of this Barnes gene? Okay. The source of the Barnes gene is Bacillus amyloliquefaciens. Bacillus amyloliquefaciens. Okay. And the most important thing, if we want to convert the sterile plant into fertile plant, we have to cross that with Barster gene. Okay. This will encode an inhibitor of ribonuclease. Because of this, fertile will be restored. Fertility will be restored. I hope this is clear for everyone. I hope this is clear for everyone. Okay. And another most important thing, in India, the BT cotton was released in 26th March 2002. This they can ask. Okay. And another important thing, India stand 5th position in the cultivation of BT crops. Okay. Cultivation of BT crops, India stands at 5th position. This is the regulatory framework how the genetically modified organism are commercialized in India. As I already told, the GEAC is the nodal organism for commercialization of genetically modified organism in India. This we have already seen. Okay. 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 Any doubt is there? With this, we are completing the transgenics in crop improvement. Any doubts? Any doubts? Any doubts? Any doubts? No, sir. Yes. If no doubts means Dikshita, no doubt. Yes. If no doubts means we can see the last topic. Okay. We are going to see about, yeah, genome editing we are going to see. After that, we are going to see omics. Okay. It's just simple topics only. We are going to see what are the key points important for exam point of view. Okay. Now we have seen what is meant by gene cloning, genetic engineering, everything we have seen. Now we are going to see about genome editing. Okay, genome editing, similar concept only. Genome editing means what? Here, target mediated modification will be there. If, for example, a gene X is there, I want to delete this gene. I will target the specific gene. Okay, so genome editing means what? Target modification of an organism DNA at specific location using engineered nucleases. Using engineered nucleases. Okay. How it will make? If the double stranded DNA is there, This engineered nuclease will make double strand break. Okay, double strand break will be make. This double strand break will be repaired by homologous end repair or non-homologous end joining. This is the two different repair mechanism. Because of this, indels will be formed. Insertion or deletion will be formed. The gene function will be disturbed. Clear? So, the gene function will be disturbed. What are the questions they will ask here means, okay, who has discovered which genome editing techniques? What are the different nucleases they will use for the different genome editing techniques? Okay. Another important question we are going to see, CRISPR-Cas. Okay, CRISPR-Cas. This technology was developed by whom? Okay, this is very important objective point. Okay, CRISPR-Cas technology was developed by Doudna and Charpentier. Doudna and Charpentier. For their work, they have received Nobel Prize in 2020 in the field of chemistry. Okay, they have received Nobel Prize, Doudna and Charpentier in 2020. Okay. Now we are going to see what are the different classes of genome editing techniques. Okay. Most important things only. Okay. Three different classes are there. First one most important thing is zinc finger nuclease. Second is talents. Third is CRISPR-Cas technology. Okay. We are going to see what is what. Okay. What are the key points for exam? Okay. You see here. Okay. Zinc finger nuclease. Okay. First most important question, what nucleus they are using in this zinc finger nuclease technique? Okay. They are using FOC1 nucleus. This is the question they asked in 2023 JRF. Okay. So FOC1 endonuclease, FOK1 endonuclease will be used in zinc finger nucleus genome editing technique. Okay. Here another most important thing is, if you see for example, I want to make a double strand break in this particular position, I can design a zinc fingers. Okay, I can design a zinc finger that will go and bind at the particular position. Okay, and the what question and what the key difference is, in zinc finger nucleus, each zinc finger means what? It's a DNA binding domain that will go and bind with three nucleotides. Okay, so it will go and bind three nucleotides at a time. Okay, but if you see in talents, this each talent will go and bind with each nucleotide. Okay, clear? This is the key difference between zinc finger nuclease and talent nuclease. These two are similar concept only. The key difference is zinc finger nuclease will bind with three nucleotide, but talents will bind with only one nucleotide. And we also the same endonuclease they are using here, FOC1 endonuclease they are using. Okay, that will create a double strand break. This double strand break will be repaired by non-homologous enjoining or homologous mediated DNA repair will be there. Because of that, the gene function will be disturbed. Okay, you have to know what is meant by talent. This expansion they can ask. Okay, talents means what? Transcription activator like effector nucleases. Okay, this definition they can ask. Okay, transcription activator like effector nucleases. Okay, then most important technique we are going to see about CRISPR-Cas. Okay, here also same thing. What is mean by CRISPR? The expansion you have to know. Clustered regularly interspaced short palindromic repeats. That is the expansion for CRISPR. What is mean by Cas? Cas means what? CRISPR associated endonuclease. Okay, this is the expansion for CRISPR-Cas. Okay, there are three major components in CRISPR-Cas genome editing. Okay, first and most important thing is there should be a guide RNA that will be around 20 base pair. Okay, that will be around 20 nucleotides length will be there. This will be complementary to the gene of interest. Okay, clear? So guide RNA will be there. That will be complementary to the gene of interest. And the second most important thing is it should have an endonuclease, that is we are using Cas9 endonuclease. And third important thing, it should have a PAM site. Okay, PAM site should be there. Okay. protospacer adjacent motif should be there. These are the different things needed. Okay. For example, I want to target the specific gene. This is gene X. Okay. I will design the guide RNA that will be complementary to this gene X. Okay. It will go and bind here. Once it bind, the Cas endonuclease will become active. It will make double strand break here. This double strand break will be repaired by HDR or non-homologous enjoining. Because of that, the gene function will be disturbed. I hope this is clear for everyone. Okay, so these are the three major genome editing technique. Okay, zinc finger nuclease, talons, and CRISPR-Cas. Okay, now we are going to see how at a transcription level or post-transcription level gene regulation can be happened. Okay, these are the most important things. Okay, here we can use epigenome editing technique. Okay, in epigenome editing technique, what they will use means they will bind the effector nucleases. Okay, so epigenome editing means what? First, what is mean by epigenomics? Okay, if a DNA sequence will be there, the DNA sequence will not be changed, but some of the modification will be there. For example, DNA methylation will be there. That may express the gene expression that will increase or decrease. So, the simple definition for epigenetic is what? Heritable change in the gene expression without changing the nucleotide sequence. Clear? The nucleotide sequence will not be changed. Okay, but the expression of the gene can be changed. Okay, so we can methylate the DNA at particular cysteine, particular cytosine residue. Because of that, the expression pattern of the particular gene can be changed. Okay, and the most important thing, this they can ask in current affairs. Sir, for example, general agriculture question they can ask. What are the recently developed, recently developed genome edited rice varieties? This I already told. Two different varieties are there. One is DRR Dan 100 that we can also call as Kamala. They have targeted here CKX gene. Cytokinin oxidase gene they have targeted. Then another most important thing, Pusa rice DST1. DST means drought and salt tolerance 1. These are the two rice varieties, that is world first two genome edited rice varieties. One for drought and salt tolerance, another for increasing the grain weight. These are the two rice varieties developed through CRISPR-Cas genome editing technique. This question they can ask this time. Any doubt is there so far? Any doubt is there? Any doubts? Any doubts? Any doubts? Yes. Kindly reply. Any doubt is there? No doubts. Yes. Now we are going to see about RNA interference. RNA interference. So far we have seen how the how mutation can be happened at gene level. Now we are going to see if a particular gene is expressed, that will produce messenger RNA. How the post transcription gene silencing can be happened. There comes the role of RNA interference. RNA interference. So RNA interference is what? It's a mechanism of post-transcriptional gene silencing. Who has discovered this one? Fire and Mello in C. elegans. This is important question. In C. elegans, Fire and Mello discovered this one. There are two different mechanisms are there. One is microRNA mediated and second is siRNA mediated. The key important thing you have to remember is miRNA means that will be always endogenous. siRNA means exogenous or endogenous, but miRNA is always endogenous. This is the key important thing you have to remember. And another important thing is the size of the miRNA. miRNA will be around 21 to 23 nucleotides will be there. siRNA will be 20 to 25 nucleotides. This is the key important point. Next most important thing is Wix. You just have to know the expansion of the Wix. Wix means virus induced gene silencing method. Okay, virus induced gene silencing method. Okay, here viral vectors will be used against the target gene, okay, to know the function of the particular gene. Okay, so virus induced gene silencing, that's why we are saying this is a specific tool for studying the function of gene, so comes under the class of functional genomics. Okay, comes under the class of functional genomics. Okay, this is another repeated slide. Okay, antisense RNA technology we will use for what purpose? For flavor saver tomato gene. So flavor saver tomato was developed by the antisense RNA technology. This is why I am saying repeatedly, means this is the key questions. These are the question areas they will ask. Okay, so you have to learn about this. Okay, last topic, omics. Okay, last topic. From this lecture, you will sure you will get minimum 15 questions. Okay, so don't skip even a single slide. Okay, these all are very, very important. Okay, very, very important concepts only we are going to revise. So be attentive in class. Okay, so omics. Okay, there are different omics technologies are there. Okay, omics means what? Omics means it's a comprehensive study of biological macromolecules. Okay, comprehensive study of biological macromolecules that collectively form the structure, function, and dynamics of an organism. Okay, there are different classes of omics are there. Okay, genomics, transcriptomics, proteomics, metabolomics, and metagenomics. Okay, here the key question what they will ask means what are the different omic technology used? For example, studying protein-protein interaction, what are the techniques they will use? Protein DNA techniques, what are the things they will use? Okay, you have to focus based on definitions, you have to focus. Apart from that, the technique, nothing else. Okay, then some of the tools, softwares, what are the softwares they will use? Okay, first genomics. Genomics, what it will do? Primarily used for studying the DNA. Transcriptomics for RNA. Proteomics, we will study proteins. Metabolomics for metaboloids. This question they have asked in JRF 2020. Okay, what is mean by metagenomics? Okay, what is mean by metagenomics? Metagenomics means what? It studies the mixed community genomes taken directly from the environment. This is the key point. Okay, taken from the environment such as soil, seawater, human gutter, wastewater. Okay, this is the key important thing. They have asked the definition. Okay, in this omics, they are asking specifically about the definition only. What is comparative genomics, structural genomics, functional genomics? Like this definitions only they are asking. So you have to read the definition properly. Okay. Okay. Before going to that, one important thing. Okay. You have to know the size of genome for each and every organism. Okay. At least for major cultivated crops, what is the size of rice genome, wheat genome? Okay. These are the key questions they can ask. Okay. What is the size of rice genome, wheat genome? In match the following, they will ask this question. So you have to remember about the genome size. Okay. So this genomics, we know genomics means study of genes, the DNAs we will study. This they have classified into structural genomics, functional genomics, comparative and metagenomics. Okay, we are going to see now what is what. What is structural genomics, functional genomics, comparative and metagenomics, and what are the major techniques they are used. Okay, what is structural genomics? Definition only. Study of physical organization, structure, and sequence of an entire genome. This is known as structural genomics. Okay. We will study the physical organization, structure, and sequence. See, sequence of entire genome also we can study here. Okay. So here different sequencing techniques we will use. This is the key point. Okay. Now we are going to see the different sequencing techniques. Sanger sequencing, Illumina, Solid, PacBio, Oxford. These are the major sequencing techniques. You have to know what is the generation, whether it's an first generation, second generation, third generation, like that. You have to know about that one also. Okay. So we have seen about what is meant by structural genomics. This definition is very important. Okay. Now coming to the point, functional genomics. Functional genomics, what? It's study of how genes and genomes functions. That is the expression of the genome, interaction on regulatory network. Here the key point is what? Interaction. expression, okay? How a protein and protein is interacting. What are the techniques we can use? How a protein and DNA is interacting. What are the techniques we can use? That is the key points they will ask. Okay. You see here, I have listed the key techniques they are used in functional genomics. This is the key point they will ask. Okay. For example, Y2H. This will be used for protein-protein interaction studies. Y2H will be used for what? Protein-protein interaction study. Then tilling means what? Targeting induced local lesion in genomes. This is important question they have asked in JRF 2020. Okay, what is the expansion of tilling? Okay, clear. So you have to focus what are the different techniques they are used. Okay, another most important thing, comparative genomics means what? Comparative genome means comparing the sequence, structure, function across different organisms to understand its evolution and gene function. Okay, simple definition, comparative genomics. Okay, this is another important point. What is orthologous gene and what is paralogous gene? Okay, orthologous gene means what? It's the same gene in different species. Okay, X and Y are different species, but it is having a same gene. Okay, if gene related by duplication within a species, that is known as paralogous. Okay, they can ask the definition of what is meant by orthologous and paralogous. Okay, they can give two different statements. Which statement is correct? Orthologous or paralogous, like that. Okay, these are the key important thing. Okay, then transcriptomics. This we have already seen. Transcriptome means what? The expression of genes, how the RNAs are produced. Okay, that we can study. Okay, study of complete set of RNA transcript produced by genome under specific condition. Okay, here the key thing what they will ask means RNA sequencing technique we can use. DNA microarray, this is the key thing they will ask. DNA microarray is specifically used for finding the transcriptomics. That means differential expression of genes between the genotype at specific condition can be studied by DNA microarray. Okay, clear? Proteomics, this is simple thing. Entire set of proteins expressed at particular stage can be studied in proteomics. Okay. This is important, most important slide. Okay. What are the different techniques they will use in proteomics? Okay. We are going to see only the techniques. 2D phage they can use, MALDI-TOF, LC-MS. This is the three different techniques used in proteomics. Okay. Then I have listed some of the post-translational modification. This we are going to see tomorrow also. Just you read once. Okay. Next, these questions they asked three times continuously. Three times they have asked. What are the different techniques used for studying protein-protein interaction? Okay. These are the four different techniques used for protein-protein interaction. Okay. Y2H, yeast dihybrid technique, co-immunoprecipitation, okay, FRET, fluorescent resonance, energy transfer, and pull down assay. These are the different techniques used for protein-protein interaction. Okay. This technique you have to know, you don't want to study about the principle. Just you know what technique is used for which thing. This is enough for JRF examination. Okay. Again, I am saying you have to know the techniques. What are the technique they are using? Okay. Then metabolomics. Metabolomics definition what? Comprehensive study of all small molecule present in biological system. That is, what are the metabolites that are expressed at particular stage at particular tissue that we can profile by using metabolomics. Okay. The key technique what they will use? The key techniques they will use is GCMS, LCMS, NMR. These are the key techniques they will use. Okay. These are the key techniques they will use in metabolomics. Okay. Those who want to solve topic wise subjective, kindly enroll in this. Okay. Our mock test series. And those who are preparing for ICAR JRF examination 2027, kindly enroll in Plant Biotechnology course. It will be highly useful for your preparation. Okay. With this, we are completing our first session. Okay, any doubts is there? Any doubt is there? And kindly like post your comments about the session also. Any improvement is needed? Okay, kindly comment your suggestion also. Okay, with this we are completing our session. Okay, I will see you all at tomorrow morning. Okay, any doubt is there? Any doubts? Any doubts? If no other doubt means I will close the session. Okay, yes, students, thank you. Morning class around 11 o'clock we will have. Okay, morning class we will have at around 11 o'clock. Kindly post your suggestion in comment section. Okay, whatever suggestion is needed. Okay, how was the class? Is there any suggestions? Everything kindly post in the chat session. Okay, thank you, students.